Yeah, so it's good evening for me and good afternoon for you all, I believe. So we're going to be talking about this paper that it's one of the major papers from my PhD. And obviously Christina's the senior author on this paper. And I think you could probably see from the dates, the first submission was October 2023, and the paper was finally accepted in March 2025, which I think gives some hint about the twists and turns of the rebuttal process that Chris is going to discuss in more detail. But I'm going to give you plenty of background on this work and the mechanism that I was studying in my PhD. It is a little bit out of order compared to the final paper, but I'm hoping everything will make sense in the end. So as background, really, diabetes it's the fastest growing global epidemic with 642 million people projected to be living with diabetes by 2040. It's a really, really enormous global problem. And we know that patients that have diabetes experience an elevated risk of heart and vascular disease, including a 2 to 4-fold higher rate of myocardial infarction with poorer survival rates post-event. And for diabetic patients with peripheral artery disease, they face an 8-fold higher rate of lower limb amputations, which is exacerbated then as well by poor cutaneous wound healing. And the real underpinning cause to all of these complications is an impairment to ischemia-driven angiogenesis, regardless of a patient's glycemic control. And although we have many drugs that we can use to treat vascular complications and diabetes, but none of these specifically target that underlying core problem of impaired angiogenesis. So therefore, we think there's a really significant unmet clinical need for new approaches to stimulate angiogenesis and improve outcomes for these patients. So we know angiogenesis is driven by endothelial cells. They proliferate and migrate in an ischemic environment to form those new vessels that help the tissue heal. And one of the essential things that an endothelial cell needs to do to actually survive in that ischemic environment is reduce its oxygen consumption. We know that endothelial cells are very metabolically plastic, and they change in response to this ischemic environment. And one way that they can do this is to suppress mitochondrial respiration, which is controlled by hopefully I can get my pointer, the PDK4 PDC axis. And PDK4, pyruvate dehydrogenase kinase 4, is a mitochondrial enzyme, and its role is to phosphorylate the pyruvate dehydrogenase complex and reduce the flow of glucose-derived pyruvate to acetyl-CoA, therefore reducing oxygen consumption and reducing mitochondrial respiration, and helping the cells survive in those low oxygen environments. We know that this mechanism is important generally for cell survival in low oxygen environments, but what we don't know is if it's important for angiogenesis or how diabetes affects this mechanism. So the real whole idea behind this study was that we hypothesized that PDK4 was really important in endothelial cell angiogenesis, and that it is also impaired in diabetes, but that we can correct this response to rescue endothelial cell function and improve wound healing. So the first thing we wanted to do is determine whether PDK4 actually does play a role in angiogenesis at all. We know it's important in hypoxia. Does it play a role in angiogenesis? So I firstly knocked down PDK4 in human coronary artery endothelial cells using siRNA, then exposed them to high glucose conditions and performed a matrigel tubulogenesis assay. And we found that, yes, high glucose exposure does decrease tubulogenesis, as we would expect. And then, quite excitingly, these cells that were deficient in PDK4 due to the siRNA also exhibited impaired tubule formation. And so this really indicated to us for the first time that this mechanism, the PDK4 PDC axis, is important for endothelial cell angiogenesis. Next, we asked, how does diabetes affect this mechanism under hypoxic conditions? So under just hypoxic conditions alone, under normal glucose conditions, we observed that the endothelial cells successfully upregulated PDK4 mRNA in response to hypoxia. They increased phosphorylation or inactivation of the PDC, and they suppressed mitochondrial respiration, which is all as we would expect. And so we measured respiration using the Seahorse bioanalyzer system. And these findings really demonstrate that, or reinforce, I suppose, the importance of this mechanism in hypoxia. Next, we asked what happens in diabetes. So when we expose the cells to high glucose, so 25 millimolar glucose, PDK4 mRNA remained just at the same level as did PDC phosphorylation. However, mitochondrial respiration was significantly increased. So this to us indicated an overstimulation of mitochondrial respiration under high glucose conditions and a failure of this mechanism to adapt. And this was also associated with significantly reduced or impaired endothelial cell tubule formation and migration. And we saw that this translated to the in vivo context as well. So I examined PDK4 expression in non-diabetic mice and diabetic mice that were rendered diabetic through the streptozotocin injection, and then underwent our diabetic wound healing surgery, where we basically create two full thickness wounds on the dorsal surface of the mice to create a sort of hypoxic wound environment. So we tracked PDK4 expression over time, and we found that PDK4 was strikingly increased in the non-diabetic mice 24 hours post-induction of that hypoxic wound environment. But in the diabetic mice, this induction was significantly impaired. Again, similar to our in vitro findings, indicating a failure of this mechanism to respond under high glucose conditions. That's clearly more important in the early stages post ischemia or hypoxia, and it does appear to be transient as well. And importantly, it was associated with a lasting impairment to neovessel development in the diabetic wounds, as demonstrated by this CD31 staining. So we've demonstrated that diabetes does impair the induction of the PDK4-PDC axis in response to hypoxia, and that it's an important mechanism in ischemia-driven angiogenesis. Next, we asked, can we rescue this impairment by modulating PDK4 expression to improve the induction of the mechanism in response to hypoxia? And that's where the reconstituted high-density lipoproteins come in. So our lab has been investigating HDL as a potential modifier of PDK4, and we've been investigating it in diabetes-impaired angiogenesis for many years. We know that independent of its effects on cholesterol efflux, it has many other beneficial effects, including that it is pro-angiogenic in an ischemic environment in diabetes. And there's several findings here that are from our lab over the years, demonstrating that HDL stimulates angiogenesis and wound healing in diabetes. But the full mechanism behind how it actually achieves that is not fully delineated, although some previous work from our group has shown that HDL augments that classical HIF-VEGF angiogenesis pathway, though I will come back to this pathway in just a minute. So this is part of the proposed mechanism of how HDL actually changes endothelial cell function. But regardless of all of this, we've asked, does PDK4 and that critical mechanism play a role in the pro-angiogenic effects of HDL? So looking at this in vitro first, I treated our endothelial cells with reconstituted HDL prior to exposing them to high glucose. And I observed that the HDL treatment enhanced both PDK4 expression and phosphorylation of the PDC. And us being able to enhance that mechanism beyond that failure of the induction under high glucose allowed the cells to restore their control over mitochondrial respiration. And that was also then associated with the rescue of high glucose impaired tubulogenesis as well. I wanted to know whether PDK4 plays an essential role in these positive effects of HDL. So we again knocked down PDK4 using siRNA and then performed our HDL and glucose treatments in vitro as normal. And we observed that while HDL was still able to rescue high glucose impaired tubulogenesis in the scrambled control treated cells, when we knocked down PDK4, this effect was completely attenuated. So this to us indicated that the PDK4-PDC axis does play an important role in the pro-angiogenic effects of HDL. finally, I tested this in our murine model of diabetic wound healing. Again, this is one of the figures that's a little bit around the other way in the paper. But in line with those findings that were conducted in vitro, we found that HDL was able to restore the induction of PDK4 in the diabetic animals immediately post-woundings in that 24-hour period, and enhance PDC phosphorylation in these wounds as well. And this was associated with a significant increase in wound blood flow measured by laser Doppler perfusion imaging in those early stages when angiogenesis is most critical in wound healing, and also a significant increase in the presence of CD31 positive neovessels in the diabetic wounds as well. And HDL also significantly enhanced wound closure in the diabetic mice, which further highlighted its potential as a treatment for chronic wounds. So coming back to one of these big questions that we had was how HDL is affecting PDK4 expression. And you remember just before I mentioned the HIF-1 alpha VEGF pathway. Well, unfortunately, in this particular study, we did not observe any increase in HIF-1 alpha expression with HDL treatment under high glucose conditions, even though we were observing decreases in the PHD. This is PHD3 here. We were observing decreases in the PHD proteins, and that was an effect that had been observed before. So sort of unable to rely, I guess, on HIF-1 alpha as the mechanism that's driving PDK4 expression, we screened a number of other relevant transcription factors before honing in on, there's no change in HIF-1 alpha despite a decrease. We honed in on FOXO1, or FORCED-FOX01 protein, which is another transcription factor that is known to increase the expression of PDK4 and has a relationship with the PHD proteins as well. So just briefly, when FOXO1 becomes phosphorylated, it is excluded from the nucleus, which reduces its transcriptional activity. And in our endothelial cells in vitro, I observed that the HDL treatment decreased FOXO1 phosphorylation, meaning that it would be more likely to remain in the nucleus of the cell and potentially increase PDK4 transcription. But to confirm that it was actually targeting or increasingly targeting PDK4 specifically, I performed a chromatin immunoprecipitation assay, which allowed me to measure how much FOXO1 was actually bound to this region of the PDK4 promoter. And I confirmed that HDL treatment did indeed increase binding of FOXO1 to the PDK4 promoter, supporting its important role in HDL's mechanism of action. So to summarize this whole study, we've demonstrated for the first time that knocking down PDK4 does significantly impair tubulogenesis. Then when the cells are exposed to high glucose, which also impairs angiogenesis, this leads to an impairment to the induction of PDK4 and phosphorylation of the PDC under hypoxic conditions, and an increase in oxygen consumption in these cells. Finally, when we restore the PDK4 expression using HDL under hypoxic conditions, this reduces cellular oxygen consumption, increases PDK4 and PDC and the PDC phosphorylation, and rescues high glucose impaired angiogenesis as well. So supporting the idea that HDL potentially through the PDK4 PDC axis is a novel and really has a lot of potential as a novel therapeutic for diabetic wound healing. But the story doesn't end there. That was all the work that I completed before we submitted this paper to ATVB. I am now going to hand over to Chris, who's going to discuss a little bit more about what happened after we submitted this work for publication. So I will now stop sharing and I will hand over to Chris.
Yep. Okay. Thanks, everyone. I'm just going to share my screen, hopefully. Yep, that's it. So just, that's not normal for me, though. Let me see. Oh, I can't see the screen, so that's not good, is it? Hang on. Okay. So can you see my screen at the moment? You can.
Yes.
I can't see it, so that's really strange.
I think you
Um,
might have to there's two tabs. One is the meeting mode, and then one is your screen. Maybe I'm not sure on the top
I've
of
not had
the.
this happen to me before. This is a new one for me in Zoom, in terms of a challenge. Yeah, that's not good. Maybe layout. Okay.
Chris, if you've got PowerPoint open and you start the
Mm,
PowerPoint and you
yeah.
can open presenter view for yourself, then you can come back to
Oh,
Zoom
gotcha.
and then
Okay, thank
share
you. I'll give that a
and
go.
then
Give that a
pick the
go.
right screen from Zoom.
Okay,
It's
that might be what I'm
pretty
doing wrong then. Okay, can you guys see my slides properly now?
Yeah, we see
no,
them in
it's
presenter mode.
Okay, then I think I can change that over. Okay, how's that?
Perfect.
Okay, great. Thanks for your patience. Right. Thank you so much for inviting us to speak about our paper today and be part of this. And thanks, Khalia, for giving the background. So my role today is to talk through the rebuttal process, and hopefully you'll find something sort of interesting or maybe helpful from kind of what we went through. So we first submitted this paper in around October of 2023. We got comments back, and the decision from the editors was that we were allowed to do a de novo resubmission, and we had a deadline of 1 year. We had 3 reviewers. They were all clearly experts in the field. We got quite a lot of comments, good and bad. And for this rebuttal, I pulled out the main ones that required relatively minor to, I guess, major experiments. So the first point of contention was that for our in vivo studies, we did only use male mice without any justification. The second point was that it was a question, so what components of reconstituted HDL were important for these stimulating effects on PDK4? The third one was, what is the importance of the scavenger receptor SRB1 on the effects, on those stimulating effects of PDK4 by reconstituted HDL? The fourth one was our reconstituted HDL modified in any way? It didn't specify the modification, but we did assume it was either oxidation or glycation. And if so, if these modifications did occur, how did that affect how HDL regulated PDK4? And finally, we were asked to determine the importance of PDK4 in the pro-angiogenic and also wound healing benefits of rHDL in vivo. So I'll go through these main points now, and all of these figures got incorporated either into the main manuscript or in the supplementary section. So the first one was, I used only male mice, and it is a great question. We have mostly focused on male mice. We know exactly how to induce hypoglycemia in the males. And when you do females, it does obviously increase its time and number. And sometimes just focusing on one species feels like a more efficient way. But I think this also opened my eyes up on the need to also do females in the future. So we spent a lot of time actually trying to optimize getting females to become hypoglycemic. They need more streptozotocin, like significantly more than the males, and fewer of them will leap over the 15 millimole concentration that we define as hypoglycemia. But we worked it out over time, and we very specifically try to look at, in these females, that HDL-induced PDK4 response. We didn't take it out the full 10 days. That would have a wound closure outcome as well. Instead, we concentrated on that earlier time frame, whereas Khalia showed was the time where PDK4 induction was changed in response to wound ischemia and also that effect of HDL. So what we found in females was that in these diabetic mice, that it was early, so just 1 day post-induction of wound ischemia through the wounding process, HDL was able to induce PDK4, although this was modest, it was non-significant, with really nothing seen at day 3. So if we looked at the mRNA levels in males, that was induced after 72 hours. Now, this early induction was also supported by our laser Doppler measures. So that measures wound blood flow perfusion as a marker of wound angiogenesis. And the way we've analysed this here is that any point above this 100% PBS line represents an improvement of wound blood flow perfusion by the addition of HDL. So we can see here in the females that HDL induces wound blood flow perfusion over the controls really early, just 24 hours after the wounding, and then it starts to decline, although yet still significant after 3 days. So this is kind of in line what we see here with the PDK4 expression in these wounds, and indicates there is some kind of sexual dimorphism where there's an earlier induction of PDK4 and wound angiogenesis in the females, and we're not entirely sure of why that is the case, but it's interesting and something we need to look at in more detail in the future. So the second question was around what components of reconstituted HDL were important for its effects on PDK4. So I'm sure many of you know, but if you don't, we actually make reconstituted HDL in the lab. We purify the apolipoprotein A1. That's the main apolipoprotein of HDL. We do that in the lab, and then we combine it with phospholipids to make a phospholipid vesicle, and that makes the reconstituted HDL. So it's really only got 2 components. We've looked at this before. We were comfortable with doing these experiments. So in our lab, we can make just phospholipid vesicles alone, and also we can add this purified apolipoprotein A1, or what we call lipid-free ApoA1. So we then incubated these various components in endothelial cell culture and found that, once again, our reconstituted HDL nicely induced PDK4, did not increase, the lipid-free ApoA1 did not increase PDK4, but the phospholipid vesicles alone did. So really suggesting that they're the component of reconstituted HDL that's driving the induction of PDK4. So the third question is regarding the role of the scavenger receptor SRB1. So HDL interacts with 3 main cell surface proteins or receptors. So there's the cholesterol transporters, ABCA1 and ABCG1, but there's also the scavenger receptor SRB1. Now, this is an interesting HDL receptor. It has the ability to both efflux or remove cholesterol from cells, as well as the ability to uptake cholesterol as well into the cell. And it has the most known downstream signaling mechanisms as well. And our previous work prior to this had shown that it was really SRB1 that had mediated the pro-angiogenic effects of HDL. And we had checked the other cell surface proteins as well, ABCA1 and G1, but it's really SRB1 that was the most important. But we had not done that with respect to PDK4. So we were able to really nicely, once again, as Khalia had also shown, we can knock down the SRB1 protein levels. That was indicated here by the Western blot and the analysis. And we see here that for our siRNA scramble controls, that reconstituted HDL once again could induce PDK4, but this did not occur in those cells where we had knocked down SRB1. So showing once again that SRB1 does appear to be important for the ability of HDL to induce PDK4. So the fourth point I wanted to that we had to address was reconstituted HDL modified? If so, how does this affect its ability to regulate PDK4? So is it modified? Yes, I guess this is possible. I just want to make clear that we make a lot of efforts in our preparation of it in the lab to prevent oxidation. We use EDTA in our dialysis of it. We remove the oxygen by using nitrogen to displace it. We store it really carefully at 4 degrees, and we're really strict about using it within a 2-week time frame. Now, the other one is glycation. At least for in vitro, we think that's kind of unlikely, as for the in vitro studies, we actually firstly pre-incubate ourselves with HDL. We then remove the HDL before we then go and incubate with high or low glucose, and then also prior to then the hypoxia or normoxia exposure. That's the way we've optimized the ability to do these relatively complicated in vitro mechanistic studies. However, I guess in vivo, where we topically add the HDL to the diabetic animals, glycation is a possibility. We had done something like this similar in the past as well. So you can glycate reconstituted HDL through incubation with a substance called methylglyoxal, and you can oxidize it just using hydrogen peroxide. And the incubation's for about 24 hours, and we do some changes in the way they run on a Kumasi gel, so indicating modification. So when we incubated these in vitro with endothelial cells, we found that reconstituted HDL, once again, elevated PDK4, but also so did the modified HDLs. So despite these modifications, we still see really robust inductions of PDK4. So the final big question was when the reviewers asked us, what is the importance of PDK4 for the pro-angiogenic and wound healing benefits of rHDL in vivo? And this was our toughest challenge and took the most time. So to address this initially, we went down the pathway that was suggested by the reviewer, which was trying siRNA, and then we also tried a lentiviral mediated hRNA transduction strategy. We tried different batch numbers and combinations, and we tried to optimize all the conditions, but at the end of it, all of this was wildly unsuccessful. We got no knockdown of PDK4 at either the gene or the protein level. And then really luckily, we discovered in the literature that a group had developed a really specific inhibitor of PDK4 called AC. They'd gone to great lengths in the manuscript to show its specificity of inhibition for PDK4 compared to the other PDCs. There's 4 of them. And it had recently become commercially available, which was extremely lucky, although we did liaise with the authors of this manuscript to get advice on doses to use in vitro and in vivo and how to solubilize it best. We also took steps to validate its activities in vitro, where we showed that the presence of the inhibitor prevented rHDL induction of in vitro tuberogenesis. We also conducted a pilot study in vivo where we topically applied it to the wounds. We looked for how it was absorbed into the wounds and the time frame for that. We also looked for its biocompatibility. So did it cause any unwanted inflammatory or cytotoxic effects? But the wounds looked very healthy, fortunately, and we found no signal for inflammation in these studies. So we then set off to do a fully powered study. These mice were all diabetic. And what we found in this wound healing study, firstly, in diabetic mice treated daily with topical reconstituted HDL, we found that it did increase the rate of wound closure here in the darker blue relative to those diabetic mice just treated with the PBS control. However, in those diabetic mice treated with reconstituted HDL, but also with this PDK4 inhibitor in the lighter blue, the wound healing benefits of reconstituted HDL were ameliorated. I want to also add in here that we added this inhibitor for just the first 5 days in the wound healing process. We did this because rather than continue it for the entire 10 days, because from our previous studies, we knew that we thought that would be long enough because those changes against wound ischemia and in response to reconstituted HDL happened earlier and not really beyond 3 days, but we went out 5 days just in case. So these effects on wound closure were also supported with our laser Doppler data, where we see that in the HDL in the diabetic mice, in males, increased wound blood flow perfusion 3 days post-wounding, as we saw previously, but in the diabetic mice treated with the PDK4 inhibitor, RHDL was unable to elevate wound blood flow perfusion. So in summary, this ended up being quite a large piece of work, did show that PDK4 is important for the wound healing benefits and its induction of wound angiogenesis in diabetes. So after a year, we resubmitted our rebuttal. It was about 9 pages, where we felt we're pretty happy that we'd responded to everything. And this time it looked a little better. So nice, we had comments like, nice job responding. But still 2 of the 3 reviewers had comments, but we could address all of these just by text, and no more experiments, fortunately, were required. But unbeknownst to us, we then needed to head into what ended up being a really extended both formatting and a statistical editor's process. And I think in the 1 year that we spent during the ATV, the rebuttal for this ATVB, they had really changed, I guess, both the formatting and statistical requirements, because there were a lot of things we were asked to do that I had not seen previously and were a little bit of a surprise. I pulled out a couple of the main ones, where we had to include every single uncropped Western blot, and I'll provide a few more details about that, as well as compare every single group, including its p-value, on every graph. So in the first of these, Khalia impressively, for this paper, completed 163 Western blots, and all of them needed to be included in a separate file in the supplementaries. We had previously submitted representative examples, but that was enough to do every single one of them. They all had to be marked with the rainbow molecular weight markers, and every single band that was analysed needed to be marked and labelled. And it was a mammoth task. And it was real credit to Khalia. Thankfully, she kept excellent records of everything she'd done and the timing and the labels. So we're extremely grateful, but it was a huge amount of work. And this is just really like a word of warning to both junior and senior scientists alike. Keep excellent records of everything you do, including your Western blot images. And the second one of these was something that I'd never done before. But anyway, the statistical review was very detailed. There were a lot of parts of it that I felt were really helpful, really made the statistical analyses more robust. But there was this other element where we needed to include every single comparison of every column in every graph and include the p-values, whether they're statistically significant or not. And I'm really just saying this to explain why the figures in our paper look the way they did. We did not choose to have them this way. And so a lot of the figures are just filled up by these lines and the p-values. And my personal opinion is that kind of dilutes the clarity of the message that we're trying to get across in these graphs. We try to overcome this by bolding in the key comparisons and p-values, which were supposed to be the main outcomes. And I think in the future, I don't exactly know what I would do differently to avoid some of that, but maybe it would be to generate graphs where there were less columns, basically, but that may not always be possible. And finally, this got accepted in March 2025 of this year. It was an 18-month process, and it was really great addition to really a decade of work where we've been trying to uncover the intracellular mechanisms of how HDL promotes angiogenesis and diabetes in response to either hypoxia or ischemia. I guess I wanted to note we even got quite a few reviewer comments about this final schematic, which I didn't expect. We had initially just really focused on the main mechanisms of the outcomes of the paper with mitochondrial metabolism and PDK4. However, the reviewers must have known our previous work and just really wanted the whole story there, where the results from the current paper were indicated with red arrows, and the previous work was sort of in a lighter sort of background kind of fashion. So, but I really think the overall outcome from those comments made a much better final schematic. So I was grateful for that. Finally, I just like to acknowledge the Vascular Research Centre, which I direct in Adelaide at the South Australian Health and Medical Research Institute. I'm also part of the University of Adelaide. I'd like to thank Khalia, obviously, for doing a fantastic PhD and really supporting us right through to the end to get this paper across the line, despite leaving to go and do medicine after her PhD. I'd also like to acknowledge other members of my team, Joanne, Victoria, Lauren, Emma and Liam, who all contributed to completing those rebuttal experiments and getting across the line. So as with any big science project, it's a full team effort. And I'd like to thank all the various funding bodies, in particular for this study, it was Diabetes South Australia and Diabetes Australia funding bodies. So thank you very much, and we'd be happy to take any questions. Thank you. I'll stop sharing now.
Thank you so much. Fantastic, fantastic work. Congratulations again on your publication. If anyone has any questions, please feel free, or you can write it in the comments, or you can go ahead and ask directly. So I invite Miron, go ahead.
Thank you so much. So, wow, this was a phenomenal effort. I cannot believe the amount of questions you get in just 1 year to finalize all this, and congratulations on all the efforts that pay back in the end. I was just wondering, because to me, one of the questions that reviewers asked, and I promise you I was not the reviewer, but in any case, one of what they asked was actually which part of the HDL components are responsible. This was one of the fair questions I would say, and I was really looking forward to your explanation, because HDL is a quite complex. It has huge proteome, microRNAs are there, and then after all this, it's just phospholipids. So if you can just maybe even speculate a little bit more about this, because it is very, to me at least, it is very strange to see that actually phospholipids, which are just like a carriers of everything, are actually having this effect, this profound effect. Thank you so much.
Leah, do you want to take that, or do you?
Oh, I know that this is something that you've found previously as well, Chris, but I totally agree. You know, it's like HDL is an incredibly complex molecule. I would just highlight that at least in our previous studies, we've been using what we call reconstituted HDL, which is, as you say, the phospholipid vesicle with the purified ApoA1, so a much, much further simplified version. And that's at least just considering the translation into a therapy, that it's a much simpler molecule. My only other comment, and maybe I'll hand over to Chris, is that I don't see lipids just as carrier molecules at all. We know they have many, many of their own biological functions. So perhaps there's some nothing that I could personally comment on, but some secret function that the PLPC lipid has on its own. But I'll hand over to Chris to maybe speak in more detail.
Yeah, I completely agree with Khalia. So I think for our specific experiments, we use this purified sort of reconstituted HDL, in which we take human blood, or it's been pooled, and we actually purify the apolipoprotein A1 so it is clean. It shouldn't be modified, except in those experiments that we did. And then we combine it with phospholipid that we buy commercially. So it's a pretty simplistic particle, and that's intentional, because it's a kind of really a scavenging particle. It picks up all sorts of things, including microRNAs. It can be modified. It can carry peroxidase 1 and have a range of experiments, and it can become dysfunctional and cause problems as well. So this little reconstituted HDL particle that we make, it is functional. It effluxes cholesterol beautifully. It'll inhibit inflammation and all of those things because it hasn't got any of those contaminants or confounding factors as part of it. Look, the phospholipid vesicles did seem to stimulate, as well as the reconstituted HDL particle itself, looking like ApoA1 doesn't do a hell of a lot. That is something that we have actually shown previously with angiogenesis, but on the whole, the combination seems to be better. So ApoA1 with the phospholipid vesicles seems to, on the whole, get the best outcome. The ApoA1 has the benefit of actually stabilizing the phospholipid vesicles, because without it, they fall apart really quickly. Doing those experiments has got its challenges because you have to use them within about 12 hours of making them before we feel confident that they haven't fallen apart. But what we have found is the phospholipid vesicles don't then translate into actually inducing angiogenesis where the reconstituted HDL does. So, yeah, and that's all for me. So, yeah.
I think we have another question from Dimitris Karasis.
Yes, so hi. This is Dimitris from Miraculum. So if I can continue this discussion, okay, phospholipids by themselves can actually induce phosphorylation of PTK4, but then SRB1 is required for this effect. So I'm a bit confused because I know that ApoA1 interacts with SRB1. So have you done this siRNA SRB1 knockout experiments with phospholipids alone and not reconstituted HDL to see whether phospholipids actually activate SRB1 by an unknown mechanism? I don't know if you know the mechanism.
Yeah, so that's definitely a bit of an anomaly. So the reconstituted HDL needs the ApoA1 to interact with SRB1, and that seemed, in our hands in those experiments, it seemed that SRB1 was important for mediating those effects. Now, so clearly the phospholipid is interacting with something else, and one of those things is S1P1. It has a receptor that interacts with phospholipid-like molecules, or I think that there's other receptors that can interact with phospholipid. It could also, unlikely, but it could also enter the cell via non-specific kind of pathways as well. I don't have the exact answer to that, though, but I agree that is a bit of an anomaly where we believe we require SRB1, but then the phospholipid alone is going off and inducing PTK4. Yeah.
Also, did you try other phospholipids or PLPC only to see whether it is a more general phenomenon, including other phospholipids as well, or this is specific for PLPC, this effect?
What is a specific phospholipid is phosphatidylcholine. Is that? Yes.
Did you try other ones? Did you try other phospholipids?
No. Okay, so no, we haven't. In the past, we have experimented with different phospholipids like DPC. It was a long time ago. It is this particular one. It's got one inositol something phosphatidylcholine. If a PLPC is thought to be, well, what we found in our hands, the most functional HDL generated. And most of those studies were actually done in terms of its anti-inflammatory effects, to be honest. Yeah.
And just a final question regarding the in vivo work. So you injected reconstituted HDL to mice, So this was done once a day for, let's say, 2 weeks. Did you do any
For 10
kinetics? Did you
days.
see how clearly this reconstituted HDL is cleared from the circulation? Did you do any, let's say, in the Western blots or something? This, I suppose it's human ApoE1, right?
Okay, Khalia, do you want to answer that? Because I'm just...
Yeah, so sorry, are you asking, yes, we're adding the HDL to the wounds across 10 days. Is your question, does
Oh,
it get into
you add it to the wounds.
circulation?
You do not inject to
No,
No
not
the vein.
topical.
injected,
We
just
just
topically
Oh, I
added.
see.
quickly
Okay.
apply it. Yeah. And
Okay.
we just do it daily because that's the method.
Okay. All right. Okay,
But
that's all
no,
from me.
we didn't check to see if, say, there was any human ApoA1 circulating in the mice. We didn't check that.
No.
Oh,
But you are right though. So in previous studies that I've done, at least in terms of atherosclerosis and things like that, we would inject the mice not every day, but every other day to try and get a therapeutic effect on plaque development or anti-inflammatory effects because it is cleared pretty quickly.
Yeah. Okay,
Within
thanks.
24 hours. Yeah.
Congratulations.
Thank you.
Thank you. We have a question.
Chris, if I might add, just is it fair to assume then ApoA1 is docking the entire HDL to the membrane through SRB1 and then gets phospholipid in closer contacts, and then phospholipids can exert its effects because it is not through SRB1? Would this be a fair question?
I mean, it is a fair question. That is a great suggestion. And I think if you saw in our final schematic, we've kind of got the phospholipid just hovering around because we didn't really know how to incorporate it. That might be part of it, but it does it without the ApoA1 as well. So I think it's going through some others, like sphingosine-1 receptor, I think it's called, or maybe some other non-specific pathway, or non-receptor dependent at least, to have some effects. But yeah, that's a great suggestion. That could be what's going on.
Any more questions, maybe?
Do we have someone by Razvan with a hand up, or is that? Oh no, that's my mouse. Sorry.
I don't see any other hands. I'm looking here.
Yeah.
Do we have any? Does anyone else have another question? Anything
I might
else?
also have I had a thought also about if you maybe thought about the reconstituted HDL being a carrier for maybe potentially angiogenic drugs also to promote healing. So could we, for example, apply it plus another drug to kind of enhance the effect, or do you see it as the only treatment, for example, for translational purposes?
So that's a great comment. We haven't really so we have thought of it. We have a microRNA that if we inhibit it, it induces angiogenesis. So packaging the antagomer of that into RHDL to sort of maybe add synergistic benefit, or at least use the HDL as the delivery pathway is something we can do. And using HDL as the sort of a nanoparticle, the carrier, has been done successfully in atherosclerosis. So people have loaded in statins, and then that has directed the statin therapy directly to the atherosclerotic plaques, and they've shown reductions in plaque macrophages and inflammation. So that is certainly something. Whether maybe we could chuck in, I don't know what, something. Maybe something that could induce VEGF-A or something like that could be a good idea as well.
I'm also wondering, now that you mentioned, if HDL does have a pro-angiogenic effect, is delivering it into the atherosclerotic plaque a good idea? Do we want to promote angiogenesis there?
Do you want to have a go at that one, Khalia? Or yeah, you go. Yeah.
Oh, I think it's definitely more in your wheelhouse. I'm not so much an atherosclerosis person as an angiogenesis person. So I think angiogenesis is just, it's great everywhere except cancer and except atherosclerosis. So maybe Chris, you can.
Oh, sure.
comment.
Okay. So yeah, so we published a paper back in, I'm going to say 2014, that showed that RHDL has multifunctional effects on angiogenesis that are context dependent. We spent a lot of time on this. So what we found was that HDL could promote angiogenesis in response to hypoxia or ischemia. So it promoted that good physiological angiogenesis that you need for wound repair and regeneration. But in contrast, it would inhibit that inflammatory-driven pathological angiogenesis that you don't want, that cause plaque near vessels or can promote aberrant kind of tumor vascularization. So we think HDL can be context dependent, and now the reason why it does this is what was it? So it was its effects on
You're thinking of Nathan's work?
No, it was HIF1 alpha, because HIF has both NF-kappa B and hypoxia response elements in its promoter region, so it can be induced in ischemia but inhibited in inflammation. So that we got it down to that being the reason for this conditional regulation by HDL. Yeah. So hopefully that made sense.
Do we have any other questions? I don't see anyone with the hand up. Okay, so if there are no further questions, I'd like to thank you both for taking time to present your work. Extremely interesting. Congratulations on your publication, and please feel free to anyone who wants to also join the network to write your email here in the comment section, so that we can add you to our mailing list. We're planning now our next journal club for November, so maybe within maybe a week or so, we can we'll have everything confirmed for November. So please feel free to add your emails for everybody who's not already on our list, so we can share with you our next event for November. And thank you again to you both for your work and for your presentation, and thanks to all for joining us. I'll see you next month.
Yeah. I'll put my name down. Yeah, thank you so much for having us. Appreciate it.
Thank you.
Thank you. Thanks.
Bye-bye.
See you, Khalia, as well. See you later. Bye.
Bye.
Bye-bye.